Hello, my name is Robert Bender, and I am an associate professor of immunology at the University of Pittsburgh. I'm excited to provide a brief overview of a recent paper that we published describing the molecular mechanisms of cross-priming during tumor immunosurveillance. For over 12 years, my laboratory has been studying the immunobiology of heat-shock proteins and the roles that they play in initiating immune responses to tumors, infectious disease, and in autoimmunity. Before we discuss the current work, it will be important to go over some of the molecules and the pathways that are involved in our current study. The left side of the cartoon depicts a schematic of CD91, a large scavenger receptor expressed by antigen-presenting cells, including macrophages and dendritic cells. It is comprised of a 515 kilodalton extracellular alpha chain, which is non-covalently bound to an 85 kilodalton beta chain with a transmembrane domain and intracellular tyrosines that can be phosphorylated for signal transduction. On the right are listed three ligands of CD91, which are molecular chaperones and members of the heat-shock protein family. These are heat-shock protein 70, heat-shock protein 90, and its paralog GP96, and calreticulin, which interact with various ligand binding domains of CD91. These heat-shock proteins are chaperones of peptides inside cells. The peptides are generated by turnover of proteins and therefore represent the antigenic fingerprint of the cell. As shown on the left side of the slide, heat-shock protein peptide complexes can be released from cells following cell death or through various active secretory pathways into the extracellular space, where they bind to CD91 on antigen-presenting cells. This binding can occur in the tumor microenvironment or in draining lymph nodes. On the right of the slide, engagement of CD91 by HSPs leads to endocytosis of the heat-shock protein peptide complexes, processing of the chaperone peptides, and ultimate presentation of peptides by MHC molecules to T cells. CD91 also initiates intracellular signaling, leading to upregulation of co-stimulatory molecules and cytokine release. These mechanisms are important for priming and activation of T cells and NK cell responses and form an efficient pathway for cross-priming. Based on our earlier work, we formed the hypothesis that the heat-shock protein CD91 pathway formed an essential conduit for cross-priming of immune responses to nascent and emerging tumors. This hypothesis was formed based on the observation that the amount of antigen that is present in these emerging tumors is minute. In the context of tumor immunosurveillance, as shown in this modified cancer immunity cycle, the heat-shock protein CD91 pathway will be important in the step shown by the red arrow, which is the transfer of antigen from tumors to antigen-presenting cells for the purposes of cross-priming. In the current study, we tested this hypothesis by creating a CD91 conditional knockout mouse with specific deletion of CD91 in dendritic cells. In our model, we established tumors in mice with methylcholanthrin, a chemical carcinogen, as shown in panel A. In B, we show the results from this experiment, where induction of tumors occurs in 85% of CD91 conditional knockout mice, while only 25% of wild-type mice develop tumors. In our positive control, 95% of RAG knockout mice, which have no TOB cells, develop tumors. In panel C, we show that tumors that developed in CD91 conditional knockout mice grew more aggressively than tumors that formed in the wild-type mice. These data indicated that CD91 was important for successful immunosurveillance of tumors. In accordance with our hypothesis, in the absence of CD91 on antigen-presenting cells, there should be a deficiency in T cell responses, the cells which are depicted in red in the slide. In this experiment, we harvested tumors from mice and examined immune infiltrates by flow cytometry. We show that at the 2-week time point, there is a decrease in T cells, as indicated by CD3 marker in panel A and NK cells in panel B, by comparing blue bars to red bars, representing tumors from CD91 conditional knockout mice and wild-type mice, respectively. These differences were absent at later time points when tumors were larger and more antigen was thus available. We then sequenced tumors derived from CD91 conditional knockout mice or from wild-type mice to determine mutational burdens and neo-epitope expression. We showed similar numbers of mutations in tumors from both mice in panel A. When only affinity of mutated peptides for MHC was considered in panels B through E, no differences in neo-epitope expression was observed in tumors from both mice. However, when we examined mutated peptides based on their differential aggregate index, or DAI, tumors from CD91 conditional knockout mice had significantly more peptides with high DAI compared to tumors from wild-type mice. The DAI, which measures the differences in affinity for MHC between the mutated and wild-type peptides, appears to be the key criteria for epitope selection in generating immune responses and immune editing of tumors. We next examined CD91 in humans and report a total of 1,282 single nucleotide variations across the entire coding sequence of the protein, including within the 4 ligand binding domains that are color-coded. In this slide, we show in panel A how each single nucleotide variation impacts the ability of HSP90 or GP96 to bind CD91 by using various algorithms and computer modeling. A negative delta delta G indicates that a particular single nucleotide variation in CD91 enhances HSP binding and vice versa. Using available TCGA data in cancer patients with lung carcinomas in panel B or melanoma in panel C, we show that CD91 single nucleotide variations that enhance HSP binding correlates with enhanced CD8 T cell infiltrates in tumors, which is an indicator of better prognosis. The reverse is also true. In conclusion, our current study provides a picture which implicates the heteroprotein CD91 pathway in tumor immunosurveillance and in priming T cell responses to tumors in humans. In our future work, we plan to explore these observations in other mouse tumor models and also to take our findings to the clinic, where we can begin to examine the modulation of CD91 expression with the purpose of enhancing immune responses to patients' tumors. Thank you for watching.